Okay, so my project is related to a glioneuronal developmental tumor, which is associated to epilepsy. These are very benign tumors. However, they are the most common tumors in young patients who undergo surgery for the control of temporal lobe epilepsy, and therefore there is a high need to finding new therapeutic options for these children and young adults. In my project, we developed a mouse model for ganglioglioma based on two molecular alterations that have been described in human tissue. These are B600E mutation. This is a missense mutation that causes an overactivation of the MAP kinase signaling pathway, which regulate cell growth and cell proliferation. And also, we use an activated form of AKT, which triggers an activation of mTOR. And both pathways are critical for oncogenesis, cell growth, cell proliferation. In our work, we combine them and we introduce them to the brain of the mice at developmental time points by using intraventricular in utero electroporation, which is a technique that allows to manipulate the brain genome during embryonic times. So when we do so, the mice develop tumors very early, actually already at postnatal stages, these mice suffer from tumors. And the tumors expand, or the tumors, let's say, they are the same until adult stages, so they remain the same, which is one of the main characteristic and main power aspects of this model, because they don't grow. They are kind of they have the same size, the same phenotype, but this is the reason why these tumors actually have a long time period or a long time window to alter the brain in another fashion. And one of them is by causing alterations in the neuronal activity and leading to epilepsy. So therefore, our project is a focus on finding therapeutic markers or the underlying mechanisms to treat both the tumor per se, but also, and most importantly, the epileptic-related aspects. And I say importantly because the surgical resection has been shown that it gives a very good outcome for the tumor, so it can be very well resected. However, these patients still have epilepsy after the tumor resection. Therefore, we think that it's more important for them to find new options for the treatment of epilepsy-associated processes.